Assalamualaikum and hello everyone. I will explain the FTIR spectrum of quercetin and identify the functional groups observed in the spectrum. Fourier transform infrared spectroscopy is an analytical technique used to identify the functional groups present in a compound. It works by measuring the absorption infrared radiation at different wave numbers, where each functional group absorbs infrared light at characteristic frequencies. Before discussing the spectrum, let's briefly look at the graph. The x-axis represents the wave number, measured in reciprocal centimeters. It indicates the energy of infrared radiation absorbed by the sample, with higher wave numbers corresponding to higher vibrational energy. The y-axis represents percent transmittance, which shows how much infrared radiation passes through the sample. When a functional group absorbs infrared radiation, the transmittance decreases, producing a downward peak in the spectrum. The broad absorption band at approximately 3,407 reciprocal centimeters correspond to the OH stretching vibration of phenolic hydroxyl groups. This broad peak is caused by strong hydrogen bonding, which is expected because quercetin contains 5 hydroxyl groups. A strong absorption peak at around 1,660 reciprocal centimeters is assigned to C double bond O stretching vibration of the conjugated ketone group. The conjugation between the carbonyl group at the aromatic ring lowers the absorption frequency compared to a normal ketone. The absorption bands between approximately 1611 and 1463 reciprocal centimeters are referring to the aromatic carbon-carbon double bond and alkene double bond stretching vibrations. These peaks confirm the presence of aromatic benzene rings and alkene in the quercetin structure. The peak near 1,382 reciprocal centimeters refers to the OH bending vibration of the phenolic hydroxyl groups. The peaks in the region of 1,319 to 1,016 reciprocal centimeters are assigned to CO stretching vibration. These absorptions originate from the phenolic CO bonds and the ether linkage present in the heterocyclic ring of quercetin. Next, the peaks between approximately 865 and 706 reciprocal centimeters refers to the aromatic CH out of plane bending vibrations. These vibrations are characteristic of substituted aromatic rings and support the presence of the benzene rings in the quercetin. Finally, the absorption between 687 to 604 reciprocal centimeters are referring to aromatic ring deformation, which are commonly observed in aromatic compounds. In conclusion, the FTIR spectrum successfully confirms the characteristic functional groups of quercetin.
Hi, my name is Afiqah binti Muhamad Arid. I will explain HNMR spectrum of quercetin. First, HNMR or proton nuclear magnetic resonance spectroscopy is an analytical technique used to identify the different types of hydrogen atoms in a molecule. It provides information about the chemical environment, number of hydrogen atoms, neighboring protons, and help confirm the molecular structure of a compound. In the spectrum, the x-axis represents the chemical shift measured in a parts per million or ppm. A higher chemical shift means the proton is more de-shielded, usually because it is close to electronegative atoms such as oxygen or involved in hydrogen bonding. A lower chemical shift indicates the proton is more shielded by surrounding electrons. Now, let's discuss the peaks observed in quercetin. The first peak appears at 12.45 ppm with a ratio 1.0 as a singlet with an integration of 1 proton, assigned to H7, which is a hydroxyl proton. It is highly de-shielded because it is formed strong intramolecular hydrogen bonding with the carbonyl group. Since it has no neighboring proton, n equals to 0, so according to the n+1 rule, it appears as a singlet. Next, the second peak, the signal at 9.40 ppm with a ratio 2.81 is also a singlet and corresponds to H6 and H4 hydroxyl proton. These hydroxyl protons are de-shielded by the electronegative oxygen atoms and do not couple with neighboring protons due to rapid proton exchange. For the third peak, the aromatic proton signals that show singlet peak appear between 6 and 8 ppm. With 2 integration of proton, the peak at 7.70 ppm with a ratio 1.96 is assigned to H10 and H1. These protons are de-shielded by the aromatic ring and nearby oxygen-containing groups, causing them to resonate downfield. The fourth peak appears at 7.54 ppm with an integration of approximately 2 protons and an integration ratio of 1.96. This peak is assigned to H9 and H2. These aromatic protons are relatively de-shielded because they are influenced by the aromatic ring, current, and nearby oxygen containing groups, causing the signal to appear downfield. The signal is reported as a quartet in the spectrum. However, chemically it is more accurately described as a doublet of doublets or a complex multiplet. This is because each proton is coupled to two non-equivalent neighboring aromatic protons, such as H8 and another adjacent proton, which have different coupling constant. Therefore, the peak appears more complex than a simple doublet. The software automatically assigns the pattern as a quartet because the overlapping aromatic couplings resemble 4 lines. However, in quercetin, there are no CH3 groups to produce a true quartet, so the signal is interpreted as a doublet of doublets or a multiplet. At fifth peak at 6.9 with a ratio 1.0, an integrate of to 1 proton is assigned as H8. It is shielded because it is further from electronegative element, and it shows doublet because it is neighboring with H2. At 6th peak at 6.41 with a ratio 0.99, an integrate to 1 proton is assigned as H3. It is shielded because it is further from electronegative element, and it shows doublet because it is neighboring with H5. At last peak at 6.25 with a ratio 0.99, and integrate to 1 proton is assigned as H5. It is shielded because it is further from electronegative element, and it shows doublet because it is neighboring with H3.
Assalamualaikum and hi everyone, my name is Zakwan Harith and I will be presenting to you on the quercetin spectrum in carbon NMR. So, quercetin consists of 15 different carbon that can be seen from 15 different peaks in the carbon NMR. Carbon that is directly attached to the electronegative atom appear further downfield, which is de-shielded, while the normal aromatic carbon appear further upfield, which is shielded. Let's take a look at each carbon. So the first carbon is carbon number 4, which is the carbonyl group in the heterocyclic ring. So the oxygen in the carbonyl group actually strongly withdraw electron from the carbon, making the carbon having low electron density. This makes the carbon highly de-shielded and appear at highest chemical shift, which is 176.28 ppm. The second carbon is the carbon number 7 in the left aromatic ring. This carbon directly attached to the hydroxyl group, where the oxygen withdraw electron from the carbon, making the carbon having low density. This low density make the carbon highly de-shielded and appear at higher chemical shift, which is the 164.34 ppm. The same thing happened for the carbon number 5 in the same aromatic ring, but it is slightly shielded than the C7 due to the stabilization of hydrogen bond between the hydroxyl group at the carbon number 5 and the carbonyl group. So it also appear at higher chemical shift, which is 161.16 ppm. Let's take a look at the carbon number 9. This carbon number 9 is directly attached to the oxygen in the heterocyclic ring. Oxygen de-shield this carbon, making it appear downfield, which is 156.58 ppm. Carbon number 13 located at the right aromatic ring, which is further away from the heterocyclic ring containing carbonyl group. So this carbon directly attached to the hydroxyl group, and oxygen de-shield this carbon, making the carbon appear at 148.14 ppm. So carbon number 2 is connected to the oxygen in the heterocyclic ring and is part of the conjugation system. So the conjugation system making the carbon number 2 having low electron density that make it appear at high chemical shift, which is 147.23 ppm, that is de-shielded. So but it is slightly shielded than the carbon number 9 because carbon number 9 is an aromatic carbon that is directly attached to the oxygen. So the oxygen mainly affect the electron density, while the conjugation system in carbon number 2 widely spread through several atoms, making it less electron deficient than the carbon number 9. Carbon number 12 is similar to carbon number 13, where it is directly attached to the hydroxy group. The oxygen will de-shield the carbon, making it appear at higher chemical shift, which is 145.5 ppm, which is meaning it is de-shielded. The carbon number 3 is the most shielded among the other carbon that is directly attached to the hydroxyl group. The reason behind this is because the conjugation from the carbonyl group is extended over several atoms, making it less electron deficient. But still, it is de-shielded because of oxygen from the hydroxyl group strongly withdraw electron density from the carbon and is appear at intermediate chemical shift, which is 136.18 ppm. Now let's take a look at carbon number 1. The carbon number 1 is located at the right aromatic ring and is a quaternary carbon that connected the aromatic ring with the heterocyclic ring. This carbon having no hydrogen, making it less shielded than the other aromatic carbon due to having low electron density, and the chemical shift appear at 122.4 ppm. Carbon number 15 is the aromatic carbon that is adjacent to the quaternary carbon. This carbon having hydrogen that provides electron density, making it more shielded than the carbon number 1. The chemical shift appear at 120.44 ppm. Carbon number 11 and carbon number 14 are aromatic carbon that having similar chemical shift. The slight difference in chemical shift is due to its position from the quaternary carbon. Since the quaternary carbon attach the aromatic ring to the heterocyclic ring containing carbonyl group, it will de-shield the carbon at the ortho position, which is carbon number 11, and the chemical shift is slightly higher, which is 116.05 ppm. On the other hand, carbon number 14 is more shielded, which is 115.48 ppm. Carbon number 10 is the bridge carbon that attaches the heterocyclic ring with the aromatic ring. This carbon having no hydrogen, making it less de-shielded than the carbon number 6 and carbon number 8. Carbon number 6 is aromatic carbon that is located in between the oxygenated carbons from the hydroxy groups. It is at ortho position to both of the carbon, making it more de-shielded than carbon number 8. So it has a slightly higher chemical shift than carbon number 8, which is 98.64 ppm. Lastly, carbon number 8. Carbon number 8 is aromatic carbon that only have one neighboring oxygenated carbon, so it is less de-shielded than the carbon number 6, making it the most de-shielded carbon in this quercetin. Now I will pass to the next presenter.
Hi, I'm Dalilah. I will explain the UV visible absorption spectrum of quercetin, a natural occurring flavonoid found in many fruits and vegetables. UV visible spectroscopy helps us understand how molecules interact with light by showing the wavelength they absorb. The spectrum acts as molecular fingerprint that provides information about the electronic structure of the compound. First, let us look at the graph. The x-axis represents wavelength in nanometer, while the y-axis represents absorbance, which indicates how strongly the molecule absorbs light. High peaks represent stronger absorption. As shown in the spectrum, quercetin displays two characteristic absorption bands of flavonoids, known as band 2 and band 1. First, the first absorption peak appears at approximately 250 nanometer and is known as band 2. This band is associated with the benzoyl system, mainly involving the airing of the flavonoid structure. The absorption occurs due to a pi-pi electronic transition, where electrons and a bonding pi orbital absorb UV light and are excited to a higher energy antibonding pi orbital. Since the airing has a smaller conjugated system, a larger amount of energy is required for this excitation. According to the equation delta E equal to hc over lambda, energy and wavelength are inversely proportional. Therefore, a larger energy gap results in a shorter wavelength, explaining the absorption around 250 nanometer. The second absorption peak occurs at approximately 375 nanometer and is known as band 1. This band originates from the cinnamon system, which involves the B ring and the conjugated C ring of the quercetin. This absorption is also mainly due to a pi-pi transition. However, this region has more extensive conjugation, allowing pi electrons to become more delocalized throughout the molecule. According to this formula, wavelength and energy gap are inversely proportional. Therefore, the longer wavelength at 375 nanometer indicates a smaller energy gap than the peak at 250 nanometer. This occurs because the CMOS system has greater conjugation, producing a batochromic or redshift. The presence of multiple hydroxyl groups in quercetin further stabilizes the excited state, contributing to this shift toward a longer wavelength. The second absorption peak occurs in conclusion. The absorption peaks at approximately 250 nanometer and 375 nanometer are characteristic of quercetin. The peak at 250 nanometer represents a higher energy pi-pi transition in the benzoyl system, while the peak at 375 nanometer represents a lower energy pi-pi transition in the more conjugated cinnamon system. The longer wavelength of band 1 indicates a smaller energy gap, resulting in a batochromic or redshift. Therefore, the UV visible spectrum provides a molecular fingerprint that confirms the highly conjugated flavonoid structure of quercetin. Now, I will pass to the next presenter.
Hello, I'm Aina Maisarah. So today, I will present the mass spectrometry analysis of quercetin. So what is actually a mass spectrometry? A mass spectrometry, or MS, is an analytical technique used to identify a compound based on its mass-to-charge ratio, also known as M/Z. In mass spectrometry, the sample is first converted into ions, and these ions are then separated according to their mass-to-charge ratio, producing a mass spectrum. From the mass spectrum, we can determine the molecular ion, identify the base peak, and study the fragmentation pattern of the compound. For this analysis, quercetin was analyzed using positive electrospray ionization mass spectrometry, or positive ESIMS. Okay, next, for question 1, I will explain about the molecular ion for quercetin. Before identifying the molecular ion, we first need to know the molecular weight of quercetin. Quercetin has a molecular formula of C15H10O7 and a molecular weight of approximately 302 Dalton. In this analysis, positive electrospray ionization mass spectrometry (ESIMS) was used. During the ionization process, one proton, H+, is added to the quercetin molecule, forming the protonated molecular ion M plus H+. As a result, the molecular weight increases by approximately 1 unit from 302 Dalton to 303 Dalton. Therefore, the expected molecular ion appears at M/Z 303. Based on the mass spectrum and the reference article, the peak at M/Z 303 was identified as the protonated molecular ion M plus H+. This peak represents the intact quercetin molecule before any fragmentation takes place. Okay, for question 2, base peak. I will explain the base peak. Okay, so how can we determine the base peak? The base peak is the tallest peak in the mass spectrum. It has the highest intensity, which means it is the most abundant ion detected during the mass spectrometry analysis. From the spectrum, the highest peak is observed at M/Z 303. Therefore, the base peak is M/Z 303, which also corresponds to the protonated molecular ion M plus H+. This shows that the protonated quercetin ion is the most stable and most abundant ion produced during ionization. Because of its high abundance, it gives the strongest signal in the mass spectrum. In this study, the molecular ion and the base peak have the same M/Z value of 303 because the protonated quercetin ion is detected as the dominant ion in the positive ESIMS spectrum. Okay, for question 2. Okay, so for question 3, 3 possible fragmentations. I will explain about the 3 possible fragmentation pathways of quercetin. After the molecular ion, M plus H+ is formed at M/Z 303, it undergoes fragmentation inside the mass spectrometer. During fragmentation, the molecular ion breaks into smaller fragment ions by losing neutral molecules such as water and carbon monoxide. These fragment ions help to confirm the structure and identity of quercetin. Okay, so for question 2. Okay, so the first fragmentation is dehydration, which is the loss of one water molecule. The protonated molecular ion, M plus H+, at M/Z 303 loses one H2O molecule, producing a fragment ion at M/Z 285. The mass difference between 303 and 285 is 18 Dalton, which is equal to the molecular weight of one water molecule. This fragmentation occurs because quercetin contains several hydroxyl groups, making the loss of water possible during fragmentation. Okay, so The second fragmentation is decarbonylation, which is the loss of one carbon monoxide molecule. The fragment ion at M/Z285 loses one CO molecule to produce another fragment ion at M/Z257. The mass difference between 285 and 257 is 28 dalton, which correspond to the molecular weight of carbon monoxide. This is known as the sequential fragmentation pathway because the ion at M/Z257 is formed from the previous fragment ion at M/Z285, not directly from the molecular ion. Lastly, the third fragmentation. The third fragmentation is the direct loss of two carbon monoxide molecules from the molecular ion. In this pathway, the protonated molecular ion at M/Z303 loses two CO molecules, producing a fragment ion at M/Z247. The total mass loss is 56 dalton because each carbon monoxide molecule has a mass of 28 dalton, so two CO molecules equal to 56 dalton. This fragmentation provides another characteristic fragment ion that helps to identify the quercetin. Based on the results, quercetin was successfully identified by its protonated molecular ion M plus H plus at M/Z303. The fragmentation pattern also matched the reported fragmentation pathway for quercetin. The fragment ions at M/Z285, 257 and 247 were formed through the loss of H2O and CO molecules. Overall, the molecular ion, base peak and fragmentation pattern agree with the published data, showing that mass spectrometry is an accurate and reliable technique for identifying quercetin.
Hi, I'm Nur Aina and will be presenting the fluorescence spectroscopy analysis of quercetin. Fluorescence is an analytical technique used to study compounds that can absorb light at a specific wavelength and emit light at a longer wavelength. In this study, fluorescence spectroscopy was used to investigate the fluorescence behavior of quercetin, which is a natural flavonoid widely found in fruits, vegetables, tea, and medicinal plants. Quercetin has a highly conjugated pi electron system and several hydroxyl groups, allowing it to absorb light and produce fluorescence emission. The main objective of this analysis is to determine the fluorescence emission peaks of quercetin and to study how different concentrations influence its fluorescence behavior. Let us first understand the two axes. The x-axis represents the emission wavelength measured in nanometer. It shows the wavelength of light emitted by quercetin after it absorbs energy from the excitation source. The y-axis represents the fluorescence intensity measured in arbitrary units. A higher fluorescence intensity means that more fluorescence is emitted at that wavelength. Methanol was selected as the solvent because quercetin has the highest solubility in methanol compared to acetone and chloroform. Quercetin dissolves more easily in methanol. The molecules are distributed more uniformly throughout the solution. This produces a clearer and more reliable fluorescence spectrum, making methanol the most suitable solvent for analysis. According to the experimental setup in the journal, the fluorescence spectrum were recorded using laser excitation at either 405 nanometer or 532 nanometer. This graph shows how the fluorescence spectrum changed when the concentration of quercetin increased. There are 4 different colored curves in the graph. Each color represents a different concentration of quercetin dissolved in methanol. The blue curve represents 160 micromolar, which is the lowest concentration used in the study. The green curve represents 620 micromolar. At these lower concentrations, the fluorescence emission is mainly observed at 505 nanometer, making it the dominant peak. The red curve represents 2.5 millimolar, while the black curve represents 10 millimolar, which is the highest concentration. As the concentration increases, the fluorescence intensity at 670 nanometer becomes much stronger and eventually becomes the dominant emission peak. This slide focuses on the emission peak at 505 nanometers. This peak is dominant at low concentration, represented by the blue curve, which is 160 micromolar, and green curve, which is 620 micromolar. At low concentrations, quercetin molecules are well dispersed in the methanol solution and have minimal interaction with each other. As a result, fluorescence emission mainly occurs at 505 nanometer, producing the highest fluorescence intensity in this wavelength region. The emission peak at 615 nanometers is known as a shoulder peak. It is not the main fluorescence peak but appears between the peaks at 505 nanometer and 670 nanometer. This shoulder peak becomes more noticeable at higher concentrations and suggests the presence of an intermediate fluorescence state. It indicates that the fluorescence behavior of quercetin is gradually changing as the molecular interactions become stronger. This slide shows the emission peak at 670 nanometers. This peak becomes dominant at high concentrations, represented by the red curve, which is 2.5 millimolar, and black curve, 10 millimolar. At higher concentrations, quercetin molecules move closer together and interact with each other, leading to molecular aggregation. This interaction increases fluorescence emission at longer wavelength, making the 670 nanometer peak the strongest peak in the spectrum. Based on the spectrum, 3 fluorescence emission peaks were identified at 505 nanometer, 615 nanometer, and 670 nanometer. Using the excitation wavelength of 405 nanometer, the corresponding state shift were calculated as 100 nanometer, 210 nanometer, and 265 nanometer. The state shift indicates that after absorbing energy, quercetin loses part of that energy through molecular relaxation before emitting fluorescence. The result demonstrates that concentration has a significant effect on fluorescence intensity. At lower concentrations, represented by the blue and green curve, the emission at 505 is the strongest. However, at high concentrations, represented by the red and black curve, the emission at 670 becomes the dominant peak. Overall, this indicates that changes in concentration influence both the intensity and the position of the fluorescence emission. Overall, the fluorescence properties of quercetin are mainly due to its highly conjugated pi-electron system and the presence of several hydroxyl groups. These structural features enable quercetin to absorb light efficiently and emit fluorescence. In conclusion, fluorescence spectroscopy is an effective analytical technique for studying the photophysical properties of quercetin. The study clearly demonstrates that both solvent selection and concentration have a significant influence on the fluorescence emission spectrum.